Hello, thank you for the invitation to present at this conference. My name is Daniela Bustos Korts, and I will present some ideas about using genotypic response surfaces to predict regional adaptation. To illustrate these ideas, I will show some applications to wheat under climate change. The ideas that I will present now are also described in the paper that was recently published in Silico Plants, if you would like to hear more details. Before we start making the predictions of genotype adaptation, it's really important to understand what is our target population of environments. In other words, what are we breeding for? So the target population of environments, or TPE, is the set of environmental conditions that future varieties are likely to experience when grown by farmers. And here we are talking about the combination of soil, weather, and agronomic management. The multi-environment trials that are usually performed by plant breeders aim at representing the TPE. They are a sample of these target growing conditions. And this sample is constructed by a number of locations, years, and levels of agronomic management. Modern plant breeding programs increasingly rely on predictive technologies. This is so central because predictive technology enables breeders to increase the scale of plant breeding programs and therefore increasing genetic gain. We already discussed that multi-environment trials are a sample of the target population of environments. In the same way, breeders have a target population of genotypes that consists of the germplasm that is available to their breeding program. A sample of those genotypes are also included in the multi-environment trials. And the phenotypic data collected for this sample of genotypes and this sample of environment is highly valuable to train models that allow breeders to predict how existing genotypes would perform in new environments. Think about locations that were not included in the testing network, and therefore increasing the resolution, the spatial resolution, for example, of the predictions. Another possibility is to predict how new varieties that were not considered in the multi-environment trials would have performed in those trials. The prediction of the genotypic responses across environments might be done with many different models, and which is the most suitable model largely depends on the structure and the amount of the genotype by environment that is present in those trials. Knowing the structure and the amount of G by E is really critical because it will define the degree of detail that we need in the model and the levels at which we need to make those predictions. One of the first questions that we need to ask ourselves is, to which extent are there groups of environments that consistently induce different genotypic response? In other words, can I group years into something that I can understand a bit better, like the Niño and Niña events, like the ENSO events in Australia, for example? Or are there specific locations that consistently induce different genotypic rankings so that I can group those locations into regions and select for adaptation to those regions? And to which extent can we predict genotypic responses from multi-environment trials? So knowing the answer to these questions for your specific plant breeding program is really important, and it will determine the degree of success of your predictions. There are different types of model that can be used. Most of them are linear mixed models that are quite popular. And here at the top, I start with the most basic model that we can have, basically, in which we have an environment main effect, a genotype main effect, and a genotype by environment interaction. Both environment and G by E parts can be decomposed in different terms that describe the contribution of locations and years. Another possibility is to describe G by E in a more continuous way. For example, by incorporating environmental covariates, think about latitude, longitude, temperature, water stress variable, etc., and to estimate genotype sensitivities. If that is the case, basically we are assuming that there is a linear response of the genotype to the environmental covariate. And that situation might work fine for some cases, but it might be too simplistic and too restrictive for cases in which there is not a unique slope, but that the response is more complex. The presentation that I am showing now will focus in this second situation, in which we will use penalized splines that increase the flexibility or the type of response that the genotype can have to the environmental gradient. Specifically, we will address the case in which we have two-dimensional splines. So we might have more than one variables that are explaining the genotypic responses. To evaluate these ideas, we simulated some data to have a simple example, and this data consisted of 156 genotypes for wheat that were simulated with the Absinthe cropping system model. Genotypes differed in three main characteristics that were related to phenology. So the first one was the vernalization requirements, the second one was photoperiod sensitivity, and the third one was earliness per se. The three of them were related to different genetic makeup, and that led to a different composition of the parameter space for the Absinthe model. These genotypes were evaluated in 13 locations in Queensland, in Australia, and were run for 39 years. The objective of these massive data sets in terms of years especially was to enable us fitting two-dimensional splines. And the question was, can we increase the resolution of the prediction of yield responses using these two-dimensional P-splines? In other words, can we, if we fit two-dimensional P-splines, can we predict for locations that were not necessarily included in the multi-environment trial network? The first thing to do, because we had such a long time span, was to check to which extent we have year-to-year variation. And Australia is exposed to some ENSO events. That means there are some years that are quite hot and dry and some that are much more mild. So we used Absinthe output with some environmental variables to group years, and we arrived at two clusters. This year cluster differed in four main characteristics. So hot and dry years had lower rainfall, higher maximum temperature, higher incidence of frost, and also lower vapor deficit. Here we see the simulated days to flowering after running Absinthe for the 156 genotypes and all the year location combinations. We see that there is a large range of flowering time, perhaps larger than what you would normally encounter in plant breeding programs, and therefore this is a sort of upper bound for the G by E that can be induced by phenology. Something that I would like to mention too is that if you see that the incidence of hot and dry versus mild year changes over time. So we have here the trials ordered from the past until the most recent one, and we see that these orange trials that correspond to hot and dry trials are becoming more and more common in the most recent years, and that's a strong indication of climate change that also coincides with other Australian researchers have observed. Talking about yield, I think here is immediately obvious that we have a lot more of variation in the means, because dry trials trials in dry years had a lower mean, but there were also many heterogeneities in the variances, which indicates the presence of strong genotype by environment interaction. Therefore, that means that this dataset is nice to evaluate our ideas. So, the yield variation that was generated by the APSIM model was used to evaluate whether two-dimensional spline models could help us predicting how each genotype would react in the whole span of latitude and longitude that was considered by the multi-environment trials. So the model looks like the one at the top, in which we have the response of the genotype as a function of years as fixed effect, and then we had a number of random terms that were related to the spline. So on one hand we had the latitude, then we had the longitude, and last we had the interaction between latitude and longitude. So this was done for all years simultaneously, like the model shown at the top. And the idea was to create a sort of emulator that would describe the most important patterns in yield across two covariates. Obviously, this approach could also be extended and by replacing the latitude and longitude by other explicit covariates like temperature or so on. Nevertheless, we already mentioned before that there were strong contrast between hot and dry year and mild years. So we thought it would make sense to fit spline surfaces that are for each genotype but are also reflecting how each genotype would react in strong and in hot and dry and in mild years. And this is why we also fitted the model below, in which we have the main effects of latitude and longitude, but then we had an interaction with the year, so we had the year-specific response surfaces. So how does it look like? Actually, we arrived to something like this. Here I'm showing only 9 examples for 9 genotypes, but we produce this kind of surface for each of the genotypes. So on the right-hand side, we have something that reflects the surface of response of each genotype as a difference between hot and dry and mild years. In other words, if a genotype has a more yellow color, it means that for that specific location, that genotype severely increased its yield in a mild year compared to a hot and dry year. So immediately we can see that there are strong differences. There are some genotypes that tend to reduce their yield, like the ones at the top right, and there are some genotypes that tend to increase their yield. But this increase is also not homogeneous for the whole region. No, there are some specific parts in the predicted region that have a higher increase. So these differences in the increase induce G by E, and they also allow us to classify locations for each pixel in the map. Basically, we can know how genotypes will react, and therefore we can use this information for classification of locations. The other nice thing of these kind of surfaces is that they allow us to predict at a much higher resolution in a very simple way. Obviously, we could also have run APSIM for each individual pixel in this figure and have a high resolution grid for all genotypes. That can be done, but that's also more time consuming in the sense that we would need to have detailed information about soil for the whole region of interest, and well, computationally it also becomes a lot more difficult. So this is a sort of compromise that allows a very fast approach and that can also be applied to multi-environment trials, because here we were using simulated yield, but obviously exactly the same model with the splines can be applied to real yield data, for example. The third nice thing, or the third nice elements that I would like to emphasize, is that this kind of approach also gets a bit closer to what could be interesting for farmers, for example, because here you are predicting how each genotype would react for specific locations that were not included in the multi-environment network, but farmers might be interested to know how a genotype would perform in their specific place. So we run this analysis for the 156 genotypes, and for each pixel in the map, we were able to check what is the ranking of genotypes, resembling a little bit what could occur in a breeding program. And we did that separately for mild years than for hot and dry years, because we observed that both types of years induce strong genotype by environment interaction. So what happened? We see that in the left-hand side, we have the clustering based on the 5 mild genotypes, and we only see 2 groups, and there are some gradients from northwest to southeast. So in general, locations more in the southeast tend to induce a different genotypic ranking than those that are in purple, more in the top or the northwest. We also observe that on the right-hand side we have the hot and dry years, and we merely see that there are 3 types of environments. So locations are inducing much more G by E, and this has to do with the fact that in hot and dry years they expose more the differences in the soil conditions, and therefore it results natural that there will be more contrast and more changes in the genotypic ranking. So this is a nice example that shows that it's important to do classification on 2 levels. On one hand, to understand the structure of the years, and second, to understand the structure of the location, because the combination of the figure A and B that I am showing here is actually defining the most complex interaction we can have, that is the genotype by location by year interaction. Obviously, when we are doing this, a natural question is, yes, but to which extent I can know which year type will happen? Well, this is why we are doing long-term simulations, and we try, and Australian researchers are also trying to focus or to study the ENSO events, because the hot and mild years that we identified here that were highly conducive to G by E did not overlap 100% with the Niño and Niña events, but there were some degree of overlap. And therefore, I think it's really important for the target population of environments to also have a sort of distribution of the environment types across time. The other thing is that here, where we are dealing with hot and dry years, but like I showed before, the incidence of dry years, hot and dry years, is becoming more and more important in the most recent years. So therefore, if we classify historical trials into these 2 categories, one strategy might be to upweight those trials that belong to the category that will become more present in the future. So that could be a strategy to prepare our breeding program for a climate change situation. To conclude, I would like to say that to better understand G by E and to design more efficient trial networks that are able to cope with climate change, it may be useful to use a combination of crop growth modelings and statistical modelings. In this example, we relied on the ABSIM crop growth model, and we generated genotypes that were varying for a number of parameters related to phenology, and we ran those simulations for 13 locations and 39 years. We also generated a number of environmental indices that allows us to describe the environmental quality, and to classify years. This data was used in statistical models to evaluate some mixed models. We also identified year types, and we realized that it was really important to classify years depending on the level of stress for heat and drought. After identifying this year type, we fitted p-spline response surfaces to describe how each genotype would react in the whole span of latitude and longitude that was relevant for these multi-environment trials. In that way, we increase the resolution of our predictions because we are able to predict for each combination of latitude and longitude, and the results of those surfaces might be relevant for variety recommendation, for example. Here we fitted these p-spline response surfaces to simulated data, but obviously the next step and the next thing we are looking forward to is to apply this methodology to real data, yield data that was collected in the variety evaluation system in Australia. The predictions at high resolution that we obtained from these p-splines allows us to classify locations into adaptation zones, because we looked at the ranking of genotypes for each combination of latitude and longitude, and that allowed us to identify groups of locations that consistently induce particular responses on the genotypes. To finalize, I would like to thank the ERTC for the funding that they provided to the INVITA project, which aims at developing a number of models that allows us to predict variety performance in Australian environments, and the INVITE project that aims at improving the variety recommendation system in Europe. I would also like to thank my co-authors Martin Boer, Karin Chenu, Banju Seng, Scott Chapman, and Fred van Uybeck for their contribution to this work. And last but not least, I would like to thank you for your attention, and I look forward to having a nice discussion with you in the plenary session.